To show you what is an enzyme, we can do a simple experiment with hydrogen peroxide. Hydrogen peroxide breaks down into water and oxygen when exposed to light. You can see oxygen bubbles start to form in the hydrogen peroxide when it is left in light for a while, but the formation of the bubbles is very slow. Now, watch as I put a piece of potato into the hydrogen peroxide. Observe how the bubbles form much faster with the potato. Potatoes contain an enzyme that makes the breakdown of hydrogen peroxide much faster. Enzymes are also found in our bodies to help in digestion. We can do a simple experiment involving starch to demonstrate digestion by enzymes. This is iodine. It can be used to test for starch. If we add iodine to a starch solution, it will turn blue-black. If we add iodine to water that does not contain starch, iodine will remain brown. Rice, bread, and potatoes are examples of food that contain starch. Here are two test tubes containing water and saliva. Saliva contains enzymes that break down starch. I am going to add starch into both test tubes and leave them in warm water that is the temperature of our body. After 30 minutes, I add iodine to both test tubes. The one with water turns blue-black, showing starch is present. The one with saliva remains brown, showing that starch is not present. The enzymes in saliva have broken it down. One way to explain how enzymes work is by the lock and key hypothesis. Here, I have a lock and a few keys. The correct keys needs to fit into the keyhole before the lock can be opened. Here are some models of an enzyme and its substrate. A substrate is something the enzyme acts on. Here, the enzyme is like the lock and the substrate is the key. The enzyme has a three-dimensional shape with an active site. The substrate has a complementary shape that can fit into the active site of the enzyme. When the substrate fits into the enzyme, it forms the enzyme-substrate complex. The substrate is then broken down and leaves the enzymes as product. At the end of the reaction, the enzyme remains the same. At low temperature, the enzymes and substrates move very slowly. They are less likely to collide with each other, so less likely to form the enzyme-substrate complex. As temperature increases, enzymes and substrates move faster. At optimum temperature, the rate of reaction is the faster. Beyond optimum temperature, the enzyme is denatured. It has lost its 3D shape and active site. The substrate can no longer fit into the active site. The reaction can no longer happen. At optimum pH, enzymes work best, but if the pH becomes too acidic or alkaline, the enzyme is denatured, so there is no reaction.